Okay students, let's start with an exercise of how one can download and decide gene or protein sequences from and various kinds of databases which we are having. So in this session, I'll just teach you an most prominently used database called as an NCBI. So let's try to Google it. So if you are very familiar with an website address, you can just type ncbi.nlm.nih.gov. That will take you to the NCBI database, or if you cannot remember that, just Google and just type NCBI. So once if you give that, the first link will be your National Center for Biotechnology information, and you can access the database. So either you can directly just type in an FTP in an HTTP address like www.ncbi.nlm.nih.gov, or you can just go to Google and just type NCBI, and then you can select the first link. So we know that NCBI stands for National Center for Biotechnology Information, and this is an homepage of our NCBI database. So this is a database which gives you this and called as a primary database, where we have plenty of options, like one can submit the sequence to the database. So if you are going to sequence a new species, or if you're going to identify one new bacterial strain, so you are going to sequence it and you are going to identify that by based upon its 16S rRNA, and then you are going to say that yeah, I got a new strain, and those can be deposited in this NCBI database. Similarly, we can retrieve the data, retrieve the strain, retrieve the gene informations or genome informations of specific bacteria from this large kind of database. Apart from this, we are having an EMBL and DDBJ. But as we are in India, these are across three continents. Just they have been located in three continents. One NCBI is in USA, and we have an EMBL for an European continent and DDBJ for our Asian continent. So the usage depends upon, and most probably when we are in India, we prefer like to use an NCBI because night times the servers will be very less. I mean, the server will be free in US, so the updation will be very less. Whereas in case of if you go for a DDBJ, if you try to access an DDBJ while you are in India, because of the timing zone, we feel little congestion in the traffic. But obviously when you are trying to download and small sequences, you cannot find the difference. When you go for a larger genomes, then you can really see that difference. So how far it works. So it's always preferable that we can use an NCBI database, and this NCBI database has been provided with various options, like we can get an assembly of any proteome, any eukaryotes or any prokaryotes or any bacteria or fungi or pathogen, whatever the desired organism which you are trying to work for, and bioprojects, biosamples, biosystems, books, clinical variations, conserved domains. So like this, we have so many different kinds of databases in which we will be focusing mainly on nucleotide database and next protein database, and maybe we can go for and genes and genomes. So when you start using this, you will be knowing what this database holds for, what are the informations which we can get. And we have an database called as of an PubChem compounds, where we can get the structures of any specific compounds. And one of the most useful databases of an PubMed, which gives you an literature references for your projects which you are looking for, or if you need to find some papers, we can just go through and PubMed, we can get our details of that. And access of an taxonomy, you can just give it can give you a lineage of an specific organism. And next is of structures, we can get an protein structures or DNA structures for any kind of genes or proteins which we are looking for. And most probably we will be using an OMIM, so it's an online Mendelian inheritance in man. It can give you details about an inherited diseases informations and genes and genomes. And apart from this, we have many other options like books. One can prefer for books to read the books which we cannot get from our library. So this is all about and various kinds of databases, and apart from that one can sign in by just going into your NCBI. When if you want to submit a new sequences, we just need to sign in. So later classes I will just teach you, once if you have an sequence with you, how we are going to submit it to an NCBI and all. And this, you can see that these are the most popularly used databases which I was listing you here. And you can see that PubMed, Bookshelves, PubMed Central, BLAST, Nucleotide, Genome, SNP, Gene, Protein, and PubChem. So these are the most popularly used databases from NCBI. And even we have an analysis tools. You can just go through the trainings and tutorials. So just whenever you have and free time, you can just surf through all the options which we are having. So now let's take an example that I just want to find out. Just let's start with an PubMed. So just click on that PubMed. So here this is an option where you can choose and just type any species which you are interested for. So let's say for an example that I want to look for an Streptococcus. We'll take an mutants. So just select Streptococcus mutants, and it will give you the literatures regarding to an Streptococcus mutans which has been published recently, wherever the Streptococcus mutants has been focused. So let's take a look like what are the different kinds of an research has been carried out. So say for example, I think maybe we can go through this. It's an antibacterial activity of philodendron bark against Streptococcus mutant. It might be some medicinal plants activity. So this was an article which has been published in Microbiology and Immunology Journal in 2020, March 20th. And this is the author informations. You can see that. So this was from Japan group. And this is the abstract of their work, and the full text can be seen at this place. So like this one can collect a literature reference. So here we can see that they have used an philodendron bark, black ginger, and found that PB, that's an philodendron bark, has a stronger effect than other extracts, and they have said that the MIC ranges of 9.8 to 312 mg per ml. So this is the work which they have carried out against an Streptococcus mutans. So like that one can get an informations like this from an PubMed. So it's very important for your projects. So if you want to collect and references, one can collect and references from this. And next, moving to come just back, and you can just go for an nucleotide. So let's check how the nucleotide sequence looks like from our NCBI. So just check a nucleotide, and again just you can type Streptococcus mutans 16S rRNA. So once you type that, it will give you an Streptococcus mutant strain 16S RNA ribosomal sequence, and apart from that other bacterial strains like Bacillus subtilis and all has been given. So just click on this Streptococcus mutant strain and it will list you the sequence. So say here it's an 16S ribosomal RNA. Someone has submitted this to an NCBI. They have identified this strain, and this has been called as often a locus or accession number. So which defines that what's an accession number of this strain. So if you remember this, if you can note down this, so one can just go through for next time. Instead of giving an Streptococcus mutant strain 16S ribosomal RNA sequence, you can just simply copy and paste this, or you can just use this number to search like NR042772. And this gives you an accession number, and this says you that how many base pairs has been present in this, that is of 1545 base pairs, and the class of molecule it's an rRNA, and the type of molecule it's whether it's a linear DNA or a circular molecule, and it's from bacteria, BCT bacteria. If it is an MAM, it's from mammals, and sometimes VAR virulence, PLN plants. Sometimes you can come across an SYN, it means a synthetic DNA molecule, PHG pages back. So it gives on fungi, FUN fungi. So this is a class that first line says you gives you and clear details about what this sequence is about. So it says that it's an accession number, how many base pairs, it's an RNA molecule, it's a linear DNA molecule, and it's from an bacteria, and when it has been first published. So it's on 12th March 2019. And the definition is of an Streptococcus mutans strain ATCC and 16S ribosomal RNA partial sequencing. And the accession number and locus will be same, and the version is 0.1. It means that one time this record has been updated. And DB link, so bioproject and project number and bioproject details. So just click on this and check what the project is about, so you can gain the information for why they have used this and what kind of project they have used this, if the authors has been provided with the details. So here you can see that the small ribosomal RNA is an useful phylogenetic marker, that we know that, and this is the project details in which they have used, and you can see that how many nucleotides, the genomic DNA, transcripts, PubMed articles, and PMC, PubMed Central articles, which we are having. So these are about the details of that, and you can see that its source is from Streptococcus mutans and its entire lineage. So kingdom to species level, genus level. So bacteria, firmicutes, bacilli, lactobacilli, orders, family, and genus Streptococcus, and the reference base and who are the authors for this, and the title and the journal is unpublished, so they didn't publish, and it was a direct submission to an NCBI. And you can see here it says on futures, location or qualifiers. So source is from 1 to 1545 base pairs, and organism name Streptococcus mutans, and molecule type is of an rRNA, and its strain, and culture collection, and type material type is of an type strain of Streptococcus mutants, and the product is of an 16S ribosomal RNA, and this is your entire sequence. So this gives you an overall idea how your 16S ribosomal RNA looks like. So if you want to use this sequence for your further works, so you can just copy this, but when we are copying, we are been ending with numbers like 161 gaps. Again, we need to edit all these things. So we have an option like FASTA. Before this, just remember that this is how your GenBank file format looks like. So this GenBank file format has been divided into 3 types. One is like an header information. So this is of an header information until here. This is of an header information which gives you the locus to the comments, and next part is of an futures information, so where it gives you more information about the sequences, and the third part is of our sequence portion, that's of an origin. So it starts from first nucleotide to the last nucleotide, and remember that this is very important, like it says an identifiers, that's an identifiers for us. So this should be observed every time when you are downloading the sequence. So if it is in short sequence, it's well and good. If your genome is very big, and sometimes just because of net loading problem, the sequence may end up here, like in this portion it can end up. So you might be feeling that the sequence download has been finished, but if you need to observe this, if this is not been seen on your screen, then just imagine like this. If the sequence is like this, then it says that the sequence is yet to be downloaded. So once if you can look at this identifiers, then it says that the entire download has been finished. So this is about our GenBank file format. So now I need to have an sequence alone. So I need to make it into an easy format, user-friendly format. So we are going to convert that into FASTA format. And just click here and it will make into an FASTA format. So remember that here all the information has been deleted and just only your sequence in FASTA format has been obtained. So this FASTA format always starts with a greater than symbol, and which will be having an first line is of an identifier, and from second line onwards the actual sequence starts. So this is about our FASTA format. So now one can get your FASTA sequence into your data, into your local systems, just by selecting this select and copy and minimize, and one can just open any one text document, right click and paste it. So you can see your sequence has been pasted here, and just go for file, save as, and remember that when you are saving this file, just use and double quotes. Either you can use in 2 or 3 formats. I'll just show you the standard format which can be used, like start with the double quotes and say like S mutant 16S rRNA sequence dot FASTA, FASTA format, or you can just make it as an FA or FAS, anything, but it should be FASTA. Either it should be FASTA or FAS or FA sequence. So let's say an example that I'm saving it as of an FAS, and just close the double quotations and save the sequence. Or one can save this as simple as text file, like double quote, no need to give double quotes, just say that Streptococcus mutant 16S rRNA sequence, and it will be saved as of an text file. So either you can save it as of an text file, or you can save it as of an FASTA format. So this is of one way. And if you feel like it's very difficult to copy this sequence, we have an option, but always I suggest, instead of copying, just go here, just check an option here, send it to. So just click on this icon and you will be having an option. The first option is choose destination, and click here file, and you have an option like download one item, and format is FASTA. So if you want, you can check various formats available. So if you just go through this, how the summary looks like, or yeah, I'll show you that. Instead of before doing that, we'll see the format, but just let's download this into an FASTA format. Like just choose the FASTA format and just say create file. So your file will be download here, and just double click that, you can open your file. Sometimes this file will not be open, so I will show you how to open those files, because already in the system I am having that option for opening that download. Just a minute. Okay, so let's say this is a place where the sequence has been downloaded. So let's just say that right click, open with. Whenever you feel an difficulty like this, just say that right click, open with, and choose app. So it should come like this. Open with, choose another app. So here, if notepad is present, you can choose notepad, otherwise just go for more apps, and you can find an notepad here and say OK, and your sequence will be opened. So you can use this sequence. So these are the two methods. Either you can copy your sequence like this, or you can just go to send to file and FASTA format. So this is how your FASTA format looks like, and here we have seen in various file formats like summary, GenBank, full format and all. So let's have a look how it looks like. See here, summary. So if you click on that, this is how your summary looks like. So it says that the first line gives you the information about that sequence, how many base pairs and what's its accession number, and GenBank format. So if you click on that, it will show you the GenBank file format, the traditional GenBank file format. So out of this, you can just go to, whenever you have time, just go through this. What are the different formats? Like we are choosing an FASTA, so just take an FASTA format, copy the sequence and save it. This is the one way. So similarly, so I was showing you like and copying an accession number. So let's say example that I'm just copying this accession number for my reference, and further if I want to use this, say for an example, I'm closing this. I'm just going to NCBI, and here I'm going to choose nucleotide, and just I'm pasting that accession number instead of giving an streptococcus mutans. So I know that this is the accession number for that. So just give an accession number and just click on search. So it will take you to the same record. Okay. So this is how we are going to access an particular sequence from NCBI. Or say for an example, if you want to access some gene like, so I'm using a streptococcus mutans and I can take some gene. Let's see what are the genes has been available for this streptococcus mutans. So if you go through that, it will give you all the informations about and streptococcus mutants. What are the information? See, just like it's a complete genome, strain NG8 complete genome, LRA1 chromosome, strain HM whole short genome sequence. Yeah, see here, signal recognition particle protein FFH gene, partial DNA sequence. It's of an 459 base pairs. So YLXM gene upstream from FFH coding gene. So this is often various informations about what are the various records has been available for your streptococcus mutant. So let's take an example. Okay, let's take an example like isocitrate dehydrogenase. So just click on this. So same, that is of an 16S RNA, and this is a specific gene. So you can see here it's an accession number and base pairs, DNA, linear, bacterial, from bacteria, and a date when it has been published, and you can see here the title. So in which publication they have given this, what is the title and who are the authors, and in which journal it has been first published, and its PubMed link, and when it has been submitted. And now you can see for the futures, like it says that what is the location and organism, molecule type is of in genomic DNA, and strain name given, and it's often gene sequence. So definitely it will be having an coding regions, and the coding region starts from 1 to 477, and gene name is called as of an ICD gene, and its function is of an catalytic activity isocitrate plus the reaction, what catalytic reaction it can do, and a note they have given, and the product which comes from this gene is an isocitrate dehydrogenase, and this is a amino acid sequence of that isocitrate dehydrogenase. So like this we can get an protein sequence, and this is of your DNA sequence for that gene. So like this one can retrieve an gene sequences from our NCBI. And same if you have this accession number, so let's take this, copy this accession number and just change here to protein and access this. Just access that accession number, you will end up with the same. See here, isocitrate dehydrogenase partial streptococcus mutants, and same its locus, number of amino acids, linear, bacteria, and when it has been published, and all other details. And this is its protein sequence. Just make it into an FASTA format and take the sequence, and you can copy the sequence for your purpose. So this is how one can access our NCBI database, and we can retrieve the nucleotides and proteins from the database. Apart from that, I will show you randomly, like you can just go for books, and you can just say for medical microbiology. So one can look for a medical microbiology, and to list out you all the books. So you can just see here we have a specific book for streptococcus pyogenesis. So from basic biology to clinical manifestations, you can go through the textbooks. Say for an example, if you want to take medical microbiology, so these are the useful resources for preparing some assignments, or reading through, for making some pictures. Say for example that I just want to have an overlook of staphylococcus. Just click on that, so it gives you the whole information. I mean the entire textbook will be there. You can just go through that. You can use these figures for your presentations if you would like to present it. So you can take these pictures, you can take some statements from this for preparing assignments and all. This will be very good resource, and for reading wherever you are, you can just go through that textbooks, you can refer to that. So this is about an books, and we have seen about an PubMed database. And next, so whenever you are having time, just go through this various kinds of an databases. Say for an example, if you come for an PubChem compound, so later in other classes I will teach you what are that. Just I want to see the structure of penicillin. Just click here, PubChem compound penicillin. So here the structure of penicillin, how it looks like. So just click on that. It will show you the penicillin structure and what's its usages, what about his history and all, how it has been identified, how the chemical looks like, and it's pretty good chemical information has been available. And this is about the structure, 2D structure of that, and this is the 3D structure of your. Just hold your left mouse and turn around. You can just zoom in and zoom out, like click this plus and minus symbol. You can just click in. I will in later classes I'll show you how to take this molecule into our own system and how we are going to work. So you can change it to a ball and stick model, this stick models, wireframe models, space filling models, what are the models you want. Just you can have an option like animate. So these are the new options which has been available in PubChem. Previously it was not, we don't have these options and all. So now recently they have added these options like animate and chemical structures, structural informations, and in chemical level ligand compound ID, accession ID, and IUPAC name of that penicillin. And these are all different forms of names which has been used in the chemistry, and which you have to remember, which you have to keep in mind that we are going to use an canonical smiles format when we go for later docking studies and all. So these are the things which we can get from that, and you can see here various informations has been available. So like chemical and physical properties of that compounds, how many, what's the molecular weight, like H bond donors, acceptors, all pretty informations will be available, and special informations, and from which companies we can get these chemical compounds. So we'll go through this slowly when we come for an PubChem session. So this is all about our NCBI database, so which we are looking for. So just whenever you are having time, just go through all these databases one by one, and just if, in case if you want to learn anything from this, just let me know through your ma'am so that I can help you out how to do this and all. And so this is about our NCBI nuclear, how to retrieve our nucleotide sequence or our gene sequences from NCBI. Similarly, I just want to give you a small exercise like find out like retrieve and gene sequences and protein sequences for what are the species which you like. Maybe like you can take some, what are the dental pathogens which you like. You can just take those sequences and you can send us. So try to remember. I'll just quickly I'll go through that. So just go for NCBI nucleotide, Streptococcus mutans 16S or RNA, and just click search. So it will list you Streptococcus ribosomal RNA. we got different strains of Streptococcus. So whatever the strain which you are looking for, you can just select and just select that the opted one, and you have to note down this. So the main thing is just download the sequence in FASTA format and you can show us so how the sequence looks like. So just go through this once and let us know how you can get it. And just remember, make an habit like whenever you are going to select a specific sequence, just note down your accession number. Just note down this accession number so that you can remember. So for further research, whenever you need that specific sequence, you can just, by just giving this accession number, you can retrieve the sequence. Okay, so I just want that you people should go through this nucleotide sequence and protein sequence information, and I need you people to submit an at least 2 genes and 2 proteins and 2 16S rRNA sequences of any bacteria of your choice. So overall, I need an 2 16S rRNA sequences with their accession numbers in FASTA format. I need in FASTA format, and I'll show you that how to get your GenBank file format too. So try to retrieve the sequence in GenBank file format as well as in NCBI. So 16S rRNA and gene and proteins. So this is of an 16S rRNA. So I'll show you that randomly all the 3 how to do that. So first is of an 16S rRNA, and this is an GenBank file format. Send as file GenBank format, create file. So that file will be created GB. So now if you try to open that, that will not open. So just go to your downloads. In downloads you have this. So right click, open with, and look for the options like more apps, notepad, open. So this is of our GenBank file format. So like this you have to take for 2 bacterial species. This is of GenBank file format for 16S RNA. Like this you have to select for gene and as well as protein. That's of in first exercise. And second exercise will be the same. Select the sequence in FASTA format. Send to file, FASTA, create file. And it will be in FASTA format. So like this again I need it for 2 bacterial species. So 16S rRNA for 2, a gene sequence for 2, and protein sequence for 2. So hope you can do this, and let me know if you have any problem in this. I can help you in this. Thank you.